Good evening, everyone. I would like to thank you all for taking out your precious time from your busy schedule. This webinar is organized by Banai, Behavioural and Neuroscience Academy of India. Banai is a not for profit organization formed in 2012 and has organized more than 200 plus programs nationally and internationally. Focus of Banai is to provide contemporary cutting edge on behavioral neuroscience. Banai is formed and runs under the guidance and chairmanship of Dr. E. Mohandas. Banai welcomes everyone and anyone who is interested to join. It gives me immense pleasure to inform you that Dr. Bhaskar Mukherjee is the moderator for today's webinar. Thank you, Dr. Bhaskar, for accepting to be the moderator for the webinar. And I would now request you to take over and start the session proceedings. Over to you, Dr. Bhaskar Mukherjee.
Yes. Hello, everyone. I am introducing myself because I don't like others introducing me, because I have no interest in knowing where my, let's say I am an audience. So I would not like to know where my speaker is working, what he or she has done, or what are the other things. I would like to know what my speaker or my moderator or my chairperson stands for. And from that idea, I have made my introduction, do a presentation or moderation or chairperson. I'm a student of psychiatry, the medical discipline, dealing with the full working of brain. I don't understand mind. I don't understand mind. Mind is a hypothetical concept, not for philosopher, for literature enthusiasts, but not for us. I am a student of molecular biology, and for me, psychiatry is actually molecular psychiatry. I love to understand and evaluate the pathophysiological basis of psychiatric symptoms, both microscopically and macroscopically, going from connectomics to genomics. And my interest areas are treatment resistance in psychiatry and diagnostic understanding of psychiatric symptoms. And my life's goal is to make psychiatry regain the master spot in medicine, a spot reserved for the medical specialty dealing with the master organ of body, namely brain. So today, I don't have much work to do, thankfully. Today, my work is to let Dr. Naren Rao have his introduction, and then I'm going to hand over the session to him. So please upload his slide. Yes. Dr. Naren Rao is presently Associate Professor of Neuroscience. But for me, he would always be that person who has done what most of us have dreamed. He has joined in Neuroscience via lateral entry at the age of 45, and after doing a PhD in his own subject choice. That is, in my opinion at least, one of the most commendable things a psychiatrist can do. His research interests are neuroimaging, and he has done fellowship there. Along with that, he is also interested in neurobiology of schizophrenia, obsessive compulsive disorder, positron emission tomography, MRI, and neurocognitive assessment. He has multiple awards, but that we already know about it. So, Dr. Rao, over to you. Please unmute yourself.
Thanks, Dr. Bhaskar. Good to see you after a long time. Yeah, I'm happy to introduce Dr. Ram Bishnoi. Today he will be speaking on one of the very interesting topic that Banai has organized in the recent past. He'll be speaking on the topic microscopy to neuroimaging, multiscale neuroscience of psychiatric disorders. I think the field now has understood that a single discipline will not be able to explain the complex functioning of brain. So the scientific field has realized that we need at multiple levels different systems working, different disciplines working, and then somebody has to synthesize them so that it makes a complete picture. So I think from molecules to neuronal level to circuitry level, from circuitry the systems level, and then once the systems results in what we call as the functions of the brain. So I hope Dr. Ram Bishnoi will be covering the entire plethora of the research, starting from the microscopy till the systemic level, that is from the neuroimaging perspective. And I'm very happy to have Dr. Ram Bishnoi here. He's the assistant professor of psychiatry and health behavior at Medical College of Georgia, Augusta University. He's also an attending psychiatrist. He's practicing over there. And he's faculty member at Augusta University. He's also a member of multiple societies, including Alzheimer's Association, Society of Biological Psychiatry. He also has written several articles in the field of dementia as part of the ADNI initiative, and I also saw several articles in the Indian journals. So we are very happy to have you, Dr. Ram. And maybe in the next 40 minutes, you will have a feast to enjoy. From the Banai, there is also a best question award. The winner will get rupees 5,000 Amazon e-voucher. I have been asked to specifically tell that the award will be given in the same session at the end of the program. So whoever has the question, please type in. Myself and Dr. Bhaskar will decide, and then the winner will be announced. So without further ado, I hand over the floor to Dr. Ram, and we will get back after he completes his lecture.
Absolutely. Thank you, Dr. Rao. Now, who's going to load my slide over there? I shared my slide.
Yeah.
Hello?
Yes, sir. You have to share your.
Oh, I have to do it again?
Yeah.
Okay. All right. Can you see my slides now?
Yes, sir.
It's visible.
All right. Okay. So I'm hoping that slides and audio is all right, and you can see me and hear me all right. But thank you so much for this opportunity. I thank you, Dr. Mukherjee and Dr. Rao, for introducing me. And I'm sure that everybody had a busy day today, and I'm not going to make it more harder by keeping it more complicated and loaded. So I'm aware of the fact that mostly the audience is consisted of the clinicians, so I'll keep it simple for the clinicians and relevant to them, at the same time emphasizing the importance of this topic for our field in general. Let me start with the case here that we commonly see in emergency room. So a patient presents to emergency room with chest pain and acute shortness of breath. You check the vitals and you see that patient is tachycardic and hypertensive. You do the electrocardiogram and see ST elevation in the anterior pericardial leads, and troponin levels are elevated. Chest X-ray is okay, and his D-dimers are fine. You diagnose that patient is having heart attack, give him nitroglycerin, and refer the patient for angiography. Now, in this patient, each of the findings and the treatment decisions you make sense because they are based on the structure and the functional understanding of heart. We know how heart works, and well, we also know like what each of those findings on the vital signs or labs or on the like imaging are telling us, and this is because of the like very well-known pathophysiology of the heart. At the same time, if we see another patient who is, say, having acute anxiety attack with a subjective sense of difficulty breathing and feeling of impending doom, you examine the patient and you see that there is tachycardia and restlessness, but electrocardiogram and lab work are unremarkable. You can also do the neuroimaging like CT scan or MRI, but you're not going to see much over there because there aren't any gross structural changes. So most of us clinicians are going to offer probably a DSM diagnosis, some pills, and probably psychotherapy too. Now, if we compare this case to our first case, we know that most of our treatment decisions here are not based on our understanding of pathophysiology. None of the treatment decisions made are based on our understanding of structure and function of the causing organ, which is brain. And I think there is now consensus among all of us that the origin of these symptoms are from the brain, but we don't know how and where or what structure of brain are implicated in these functions. And uh, there's no way to target those structural or functional changes with our treatments. So there's nothing new in that. We all know that, and there's no learning point in that. But the question is, what is it going to take to us to formulate this case in the same way as the first case? What do we need to know, or when are we going to have our formulations similar to first case? And for that reason, we need to understand how brain works and what's the correlation between structure and function of the brain. And to understand any biological process, there have to be a deeper understanding about the relationship between structure and function. The brain, we know that it's made of a lot of cell membranes, mostly made of fat and water with salt and protein molecules. And brain is responsible for behaviors, for emotions, and cognitive functions. Now, we do not know how they are related, like how those structures eventually lead to these functions. And the branch of science which studied this is called neuroscience. And this is not a new field of study. It's been around for over a century. But there has been immense focus on neuroscience in the last 20, 30 years. And that's for a reason, because there have been some incredible advances in this field. Now, there are different kinds of neuroscientists based on the functions they are studying in the brain. People who study the cognition and the origin of cognition in the brain call themselves cognitive neuroscientists. Similarly, people who work on the molecular level, they can call themselves molecular neuroscientists. And then some of them are building computational models of neuroscience to understand the brain organization. And so these are not clearly defined subspecialties, but it's good to know that what they are telling us about is like their specific area of work in their research. Now, talking of the clinical neuroscience, like most medical sciences, neuroscience also have clinical specialty where the research findings are eventually translated into patient care. And the psychiatry and neurology are the important fields who are and who will be going to use all the advances from the neuroscience research. Now, as a clinical neuroscientists, we all psychiatrists need to know neuroscience, just as the surgeons need to know anatomy. I mean, some people may argue that neuroscience hasn't made any significant differences in our clinical practice or in our ability to diagnose psychiatric patients. Uh, that's true so far, but the way the way the neuroscience overall is advancing, uh, we are sure that in near future we are going to use basic neuroscience findings in our clinical practice too. So like I said, if surgeons need to know the anatomy, we clinical neuroscientists or psychiatrists need to know the basic neuroscience. Now, that was about the neuroscience. And like I said, our topic was the multi-scale neuroscience. So the question is, what is multi-scale? We know that the brain is made of billions of neurons, and that those neurons connect or talk to each other through synapses. And there are trillions of synapses. So if we look at the structural level at the cellular or subcellular level, like the cell neuronal level, or the components of neurons like the axons or even the spines on the dendrites, or genetic expression levels, that's microscale. So microscale is something which we can see under the microscope, or at the molecular level. And most of these studies are done in the animals or postmortem brain, because it's not easy to get a living brain under the microscope. Then the mesoscale is the studies where there are cellular columns of, say, about 100 to 500 cells at a time, which are packed together and have similar functions, and they connect to another group of cells and lead to a specific function. Mesoscale studies are mostly limited to animals. There are no good way to study the mesoscale neuroscience studies in human beings. And the macro scale or large scale is what we see through our modern neuroimaging methods. So the macro scale studies are the most commonly used studies these days to study the living brain inside the human body. These are non-invasive, and now with the technological advancement, they are becoming more and more useful in understanding how we understand the neuroscience and explain the behaviors and the psychiatric disorders. So overall, when we talk of multi-scale neuroscience, we are basically talking about either micro scale or macro scale, and their role in the understanding of brain in health and disease. Now, that was about the topic. So that's what I'm going to discuss further. But first, let me start with the brief history of neuroscience, how we reached to the point where we are now. And let me take you long back before Christ. So Hippocrates and Aristotle were like contemplating the philosophical view of brain, and the question there was, what was the real function of brain? And Hippocrates thought that brain was responsible for thinking and emotions, but Aristotle thought heart was the seat of the emotions and the brain was just a radiator to cool the blood. So there was this philosophical question over there, that what's the real function of the brain? Then a very famous physician, Galen, who was treating the Roman gladiators after their fights, he saw that some brain trauma were leading to the functional deficit. So he imagined the functional role of brain subregions, that certain regions are responsible for certain functions. And then there were arguments going back and forth about the real function of the brain. Around 10 or 1000 AD, Muslim physicians, Al-Zahrawi and Ibn Sina, they were the prominent thinkers of the time in the field of medicine. And Al-Zahrawi's textbook, Kitab al-Tashrif, was a main textbook for medicine for next 400-500 years. They looked at the anatomy of the over and whole body anatomy, and also the skull and the brain anatomy, and had influence on this field for several hundred years. In 600 years, Rene Descartes had this new idea that the brain and mind are different. So he proposed this idea that the mind's function is to think and feel and believe, and has nothing to do with the brain as we see structurally. That was again fraught with like a lot of arguments. And in mid 1800s, Bell, Florence, Broca, Wernicke, they all looked at the localized brain deficits, like loss of speech or loss of hearing. And then they tried to look at the brain to find the specific brain regions responsible for those functions. And they were quite successful in locating some of the brain functions over there. But at that, until then, there was a very limited idea about how brain looked at the cellular level. The most prevalent theory at the time was the reticular theory of brain, and which was that the brain is just a string which is just put together, and it's sitting inside our skull. Most people believed then that unlike heart or liver or kidney, which are made of the basic unit at the cellular level of single cell, brain wasn't like that, and brain was differently organized. Now, the next advancement in the field of neuroscience was attributed to these two gentlemen. The left is Camillo Golgi, and an Italian physician, and the right is Santiago Ramon y Cajal. Golgi developed the silver nitrate stain, which stained only the brain tissue and didn't give color to the other tissue. But he also believed in the reticular theory of brain. So he believed that it's just staining that the brain strings, but there were no cells. Santiago Ramon y Cajal was a brilliant neuroscientist, or actually a physician, and he just locked himself in a room with a simple it was a microscope, not even electron microscope by then, but a simple microscope, and animal brains, and also postmortem brains. And he just spent a lot of time looking at these brains, and he proposed that unlike the prevalent view then, the brain was also made of single cells called neurons. And he proposed this neural doctrine of brain. Even Camillo Golgi didn't agree with the Ramon y Cajal, but because of their combined work together, they were given this Nobel Prize in 1906 for the discoveries related to brain. But for the fact that Ramon y Cajal was actually the first one who proposed the structure of brain the way we understand now, and not surprising that he's called the father of modern neuroscience. So that's the beginning of modern neuroscience. Now let's see how we have progressed. Oh yeah, so let me take you back to medical school. If you remember the medical school textbooks, you have seen these pictures there, and this is the brilliance of Ramon y Cajal. These are hand-drawn pictures of the brain and organization of brain cells, mostly from animal brains. And these pictures are still used in textbooks for understanding or for teaching how brain is organized. So this is extraordinary. Now, 1889 was the year when Cajal proposed the neuron doctrine, or said that the brain is made of basic cellular unit called neurons. Interestingly, that was the time when there were a lot of other active researchers and physicians who were working in this field, and you must have heard these names like Alzheimer's, Huntington's, Freud, Kraepelin. They were all contemporaries of Cajal, and Cajal's work was not well received by most of these people, but once it was recognized and he was given Nobel Prize, people started focusing on, or they took his work further and tried to find out more. So like I said, Cajal had to work with just the microscope. There were not even X-rays. X-rays came into medical use in late 1890s. In 1906, Sherrington looked at the way these neurons communicated with each other and led to the concept of synapse. And then he again was given Nobel Prize later. So that has Cajal and Sherrington's work led to the origin of modern neuroscience. We can say that. Then Brodmann looked at the cellular structures and divided the whole brain into 52 different cortical areas based on their functional specialization. 1930s was the year when the EEG and electron microscope were discovered, and electron microscope has a huge impact on the further research because the researchers could see brain with better resolution. I'm so sorry, there's a lot of noise going on because my office is right next to a conference room, and there's a conference going on over there. Okay, 1930s, limbic circuit, Pepe's limbic circuit was discovered, and this was the beginning when people were thinking of the circuits in the brain that and they were looking for the structurally connected discrete brain regions. 1950s, Hebb and Lashley proposed this theoretical ideas of learning and memory, and the Hebbian concept of cells that fire together, wire together, still stands true. And even though they didn't have tools to look at those, look at the real, the functional aspect of the brain then, but those theoretical ideas had a huge impact later on. Then 1960s, Eccles, Hodgkin, Huxley, they proposed the cellular membrane mechanisms in neuronal. So, and then the opioid receptors in 70s. So if you look at the overall pattern, it's like zooming in from the above, like first the cell and then the synapses and then the cellular mechanisms of the membranes and then the receptor level. So we were going deeper and deeper into the brain tissues. 70s, 72 was the one when the first CT scan came into medical use, and 76 or 77 was the first CT scan study in psychiatric patients in chronic schizophrenic people. And that showed that there were dilated ventricles, and we still use that knowledge in our explanation of schizophrenia. But there has been a lot of advancement since then. 74, PET scan came into use. But the real advancement in neuroscience was in 1990s when fMRI and diffusion weighted imaging were discovered. And these are the tools now most commonly used in brain structure studies or brain function studies, actually. And these tools have made significant progress over the last 20, 30 years. And that's why the neuroscience has been in the news, and the kind of emphasis it's getting, it's because of the findings from fMRI and diffusion weighted imaging. In 2009, after the success of Human Genome Project, Human Connectome Project was launched, and the idea was to map the complete structural and functional connections in the brain. And then based on the ongoing research findings and these aspirations of neuroscientists to understand the brain better, human brain mapping project was launched by European Union and brain initiative by Obama White House. And the goal was to develop newer technology and newer research methods to understand the brain completely, or to develop the complete brain map, just like human genome. Now, at microscale level or at the cellular level, when we see the cells under microscope and what we see, these studies have been going on for centuries now, from Cajal and even before that. And look at the way those microscopes have advanced from basic microscope to the picture you see on right side is from storm microscope or optic microscope. And now you can even see those small dendrites on the spine with a great clarity. So the microscale studies have made significant advancement in terms of resolution, but the concept at the microscale level is still of functional specialization and functional integration. And that means that the cells which are common with their functions are connected to each other and have similar structures. And we see that there are pyramidal cells in the different layer of cortex, and the kind of cells or the structure of those pyramidal cells is dependent on the kind of functions they share. So the overall studies in the neuroscience have went from like regionalism to connectionism, and now we know from microscale study as well as macro scale that the connections or neural circuits are at the heart of all brain functions. And so that's about microscale study. Now, because microscale studies have limitations that we can only get the brain after death, or we can look at the animal brain. It's really hard to look at the living brain cells and look at their structure and function or changes while performing the function or task. For those reasons, macro scale studies have been the most important tool to understand brain right now. For structural understanding, we have diffusion weighted imaging or diffusion tensor imaging. And what they do is they look at the diffusion of water molecules. So like we know, the white matter is made of the white matter tracts, and they are the axons of the neurons sitting in the cortex somewhere or in subcortical structures. So those axons are like pipes filled with the water. Now, the diffusion of water molecule is limited by the myelin sheaths or the cellular membranes. So they can diffuse easily along the axis of that long pipe, but their movement is restricted in the other directions by those myelin sheaths. So this anisotropic diffusion can be measured by the MRI, and can the voxel-wise data from those fibers can be used to reconstruct the whole white matter tract. So it's easier now to understand the extent and the organization of white matter tract using diffusion weighted imaging. To understand the functions, we have EEG and MEG, and which are there for some time now, but they are good with their temporal resolution, but not spatial resolution. We don't know where those signals are coming from in the brain. PET scan creates images by tracking radioactive molecules, like injecting radio tracers like dopamine, and then looking at where this dopamine is going and what kind of effects it's having on the specific function. And the functional MRI is one of the very commonly and widely used tool at present, and it is dependent on this assumption that the brain structure which is active in performing tasks leads to the increase in the blood flow to that area, and the oxygenated and deoxygenated hemoglobin have different magnetic properties. So these differences in the magnetic properties can be recorded and can be reproduced in a more visible form, like a color blobs in the brain. And the concept of connectivity with fMRI is that the regions which show similar activity levels, like the regions while performing a task show hyperactivation at the same time, are called functionally connected regions. So even though it's indirect, but it has a significant overlap with the structural images, and this has good reproducibility. So we can confidently say that what functional MRI measures is actually the involvement of those regions in performing that functions. Now, fMRI can be done while performing tasks, like you can give a task, a memory task to patient, and record the activity at the same level, and you can see, like, okay, these are the regions involved in this tasks. But another important observation is that even at rest, when we are not performing any tasks, or when the patient is just sitting inside the scan and is not actively involved in any cognitive or behavioral tasks, even then the activity inside the brain make a lot of sense, and that have been analyzed, and that's called resting state fMRI. And resting state fMRI has been like increasingly used as a research method because there's less variability which is related to the effort or performance of task, and you are basically taking out the effects or the variance from the task performance from the results. So now these are the common tools which we use in assessing connectivity, specifically the diffusion weighted imaging and the fMRI. And what we see on the results are these images. So left one is the example of structural connectivity, and different colors indicate whether those fibers are anteroposterior or left to right. We do not really know the directions of the fibers in there, and that's one of the limitations. We don't know it's going from the, like, where is the origin and where's the end of those fibers. But again, I mean, what we see is pretty amazing. In functional fMRI, on the right side, we see the BOLD signal, or blood oxygen level dependent signal. Like I said, blood oxygen or oxygenated hemoglobin has different properties than deoxygenated hemoglobin, and that can be measured and can be used to see the activity visually. And you see the red spots are hyperactive areas and the blue spots are hypoactive areas. And again, like I said, this connectivity in the functional fMRI is basically statistical covariance. So those areas which are active at the same time are considered functional connectivity. Now, even though there is a significant overlap between structural and functional connectivity, it's not a total overlap. And the reason may be, one of the proposed reason is that some of the functionally connected regions are connected indirectly, like they're mediated by a third connecting point. But again, there is a huge overlap between structural and functional connectivity, and the reproducibility of all these fMRI and structural studies are getting better. Now, once we have these data from the whole brain, the next step in understanding the brain organization is how to assess the connectivity. And there are different ways to look at the connectivity. So the data which we have from the scan is 4-dimensional, especially in the fMRI, where it's a time series data and like 3 spatial scales. So one of the ways to look at the connectivity is just pick one seed or region based on the theory or based on data analysis. If you find a specific region which is implicated and you want to look at the connectivity of that region throughout the brain, then that's called seed-based connectivity. And again, I mean, it's important to understand the specific region, but then again, you're leaving out a huge data set in the brain. And like I said, the concept in connectivity or in the brain connectivity, as we know now, is that the brain functions of any specific region is not performed in isolation, but it's basically the interactions among different regions. So for those regions, the whole brain scan analysis or whole brain data analysis are more popular these days, and independent component analysis and the graph theories take into account all the data points in the brain. Independent component analysis is just a method of, it's a computational method of separating multivariate data into subcomponents. For example, you want to identify minimum components which can explain the maximum variance in the data set. That has led to some important and exciting finding. I'm coming to that. And then there's a graph theory, and I'm going to discuss that again in a little bit. So this is what we see from independent component analysis. And these networks are derived from the resting state network. So what was done was when the individuals were sitting inside the scans before performing cognitive tasks, they were just thinking about themselves or not doing any executive tasks or cognitive tasks, I'm sorry. And when people analyzed those data, when they were not actively involved in any tasks, even then they show that there were some regions who were showing different activities. And that led to this concept of resting state networks. And these are a few of the resting state networks which are commonly described. So when a person is laying down in a scan and not actively performing any memory or cognitive tasks or any other specific fMRI paradigm tasks, he is likely to he's likely to think about his own self, or what we call as self-referential thinking. And at that point, there are distributed brain areas which are hyperactive. And those areas work as a network, and that network is called default mode network. And as soon as they are involved in a task which involve focus and concentration, the default mode network areas, they become hypoactive or less active. And then there are different regions which show hyperactivations, and those areas form a network called executive network or central executive network. Now, there has been a huge deal of progress in the resting state brain network research. And there is another network which is anchored by the anterior cingulate and insula called salience network. And now we know that all these networks, they also have a specific organizational control. And that's in the way that salience network is kind of switch. So a person who is at rest and is involved in self-referential thinking shows the hyperactivation in default mode network. And as soon as he is involved in the tasks, he goes from default mode network into hyperactivation in the executive network. And the salience network is the one which act as a switch. So it switches the predominant activity from one to the next network. And these has been shown, the network changes have been seen in the psychiatric disorder too. For example, in depression, people involve or spend a lot of time in thinking about themselves and feeling guilty about their past. And we know that depression shows significant hyperactivation in the default mode network. And for them, salience network is not that efficient to switch them from default mode network to central executive network. And that's why they have this limited ability to focus on the tasks, and as soon as they get a chance, they will switch back to the default mode activity. So all these resting state networks are providing a great information about how our brain is organized and works, and that may have huge implications in psychiatric disorders as we understand them better and better. Now, most of these studies done are at the population level. Eventually, the goal is to get a specific finding at individual level and then use them in the clinical practice. So that's a work in progress for now. Now, looking at the brain networks using connectivity methods is a challenge. And the reason is because the brain data is very complex. And you need not only data, but specific tools to make sense of that data set. And for that, graph theory is one of the commonly used tools. And the study of brain networks and graph theory together has led to this relatively new field called connectomics. And I'm sure you've heard of other words with suffix omics, like proteomics or genomics, metabolomics. And the omics denotes, the suffix omics denotes that you study all the elements of a system. Traditionally, the research has been very descriptive, and that is that you look at, if you see elevated level of, say, protein X in a particular disease, you want to see whether this protein is implicated in that disease or no. What you do is you enroll a number of subjects and then measure the protein, and then you answer that questions whether this is involved, yes or no, and your results will prove that. Instead of choosing one protein, if you collect all the possible proteins from the system, and then look at all of them at the same time and their interactions among each other, and then you use those understandings in the identifying risk factors or diagnosis or treatment, that's called omics. So connectomics is concerned with the comprehensive map of all neural connections, and the connectome is that map which we are focused on. So connectome and connectomics is this new age neuroscience subfield, which is looking at all the possible connections and their role in the different functions in the healthy individuals as well as different disorders. So Human Connectome Project and all the newer initiative by US and European Union, Human Brain Map Project and Brain Initiative, they're all focused on understanding this connectomics. And the connectomics involves understanding the organization at the whole brain level. So from a complex data set such as whole brain, we need a sophisticated method that can help us define those networks and help us quantify them. And graph theory is one of the mathematical concepts to understand very complicated metrics data. And this, the use of graph theory has been is more commonly used these days in the whole brain resting state fMRI data to understand the structure of brain in terms of functional organizations. So graph is not, or graph theory is not only used in the neuroscience, it's been used in many other sciences to understand the complex relationship like computer science, physics, chemistry, even the social networks, social media networks, proteins or protein networks, gene networks, and all that. So let me tell you a little bit about how these graph theories tell us anything about the structure or the organization of the brain. So this is oversimplification of the whole brain data. Let's see if we let's assume that we have this data set from the functional resting state functional fMRI from the brain, and then the brain can be parcellated or divided into different brain regions based on the established atlases or anatomical characteristics, or even randomly. So the regions which we select are called nodes. And if we look at each node's relationship with every other node in the brain, and then we go from there. So the relationship between, or the statistical relationship between the two nodes is called edge. So graph theory results in these edge and node networks, and which provide a great deal of information about the organization in that system. So the starting point is, say, null model. So we assume that every node is connected with every node. And then the next step is we threshold them, or we look at only the significant connections based on their functional connectivity, or we can say the statistical correlations. And that will give us these maps, which are not random, or it's not like that every node is connected to everyone. And so what we see with the brain is this kind of structure. And if you look carefully, this will come as like this. So there are certain nodes which are densely connected with the surrounding nodes. And then, so those are the red colored nodes here. And these red nodes are also connected to other nodes. So these nodes, which are colored red here, are called hubs. And the hubs are connected with different hubs, and each hub forms a discrete area which may have functional specialization. So that's called modularity. So this is the modular structure of brain. And also what you see is that this kind of structure is because the way it's structured is because it enhances the efficacy of the network. And it's not only seen in the brain, it has been seen everywhere. For example, in the transportation system, or in the power grids, or in the gene networks, protein networks. All complex biological systems have this network property, and it's called small world property. So I'm sure you've heard of this term, small worldness, and this makes any network more efficient. I mean, it's easy to understand if we take an example of transport system. So in order to travel from here, I'm sitting in Augusta, and if I have to go to, say, Jodhpur in Rajasthan, having a direct flight from Augusta to Jodhpur will not be a very efficient way of transportation, but I will go to the nearest hub, which is, say, Atlanta, and from Atlanta to Delhi, the another hub, and Delhi to the Jodhpur. And this makes this transportation system more efficient, and it saves a lot of energy too. So the brain, because it has to sit in a limited space, has to be very efficient. And for that, it has to use the space by making those networks highly efficient. And the small world property make them really, really efficient. And we have seen that these, if you look at there are different parameters which can quantify like this average path length or the clustering or the modularity coefficient and all these. And they are different in different psychiatric disorders. So in the next presentation, I'm going to talk about an example using all these findings, and that will make more sense then. But the point is that these graph theory networks tells us about how brain is organized in healthy individuals, and with the disease, how these organizations get disrupted. Now, while there is ample evidence that the micro scale features are the key factors in systems organization, and our knowledge about how these different scale of organization interacts with each other, how those cellular changes or structure in the cell at microscopic level leads to the changes at the brain circuit level or brain network level or say organizational level is very limited. And Human Connectome project is attempting to integrate micro scale and macro scale studies together. And so going back to our case of anxiety attack patient, if we have to have a full explanation of pathophysiology, we have to understand the neuroscience at micro scale as well as macro scale level. And the researchers are now paying more and more attention to this multi-scale neuroscience, where macro scale networks from the MRI are compared with the histological histology of those regions. And the results have, although the studies are very limited, but results have shown remarkable overlap. So there are a few explanations for that. One of them is that the brain is like a Lego set, where each Lego piece, when put together, they form the large structure. But structurally, the micro scale feature is just the miniature version of the macro scale features. But that's not always true in several other systems where micro scale features are not at all in sync with macro scale features. For example, in economics, so where micro scale economics and macro scale economics has no common organizational principles. Another argument is that the macro scale organization might shape the spatial and temporal patterns of micro scale processes. And for example, there is an infectious model of Alzheimer's disease which propose that the disease process which start in the medial temporal lobe progresses over to the different network areas of default mode network and eventually involves the whole brain. And if you look at the spread of the amyloid and the tau protein, it follows a specific pattern. So that's another explanation that the macro scale organization actually decide how the cells are structured. But again, like I said, these macro scale and micro scale or interscale comparisons are still very limited and need more understanding. But coming back to our case, ideally we aim to see the precise changes in different scales from cell to the brain network. And we still have a long way to go, and there are a lot of challenges. So, and talking of challenges, this multi-scale neuroscience is a relatively new field. All the studies where they have integrated different scales in the same patient group or the research subjects, is being done over last 4 or 5 years only. And then there is this dynamic nature of neuroscience. The neuroscience has a different temporal scale as well, like the before and during and after the performance of tasks. There may be different organizations, or the brain organization may be different, or the organizational changes, I'm sorry, with the age, like in childhood to adolescence to uh, old age, there may be a significant change in neuroscience. So this dynamic change, and then the changes because of learning of new skills, um, uh, or acquiring more knowledge may change the organization significantly. And then another challenge is the methods and data. So neuroscience, or especially the network neuroscience or connectomics, is still in infancy, and different researchers and research groups have their own sets of methods and data, and reproducibility of the research is a significant challenge with the neuroscience overall at this time. Now, another one is, although we are using a lot of data set now, but looking at the complexity of the brain, the big data we have is not so big yet. And then the chronological change, or because of the dynamic nature and the disease effects, it's not clear like how the brain is organized before the onset of disease and during the early part of disease, and with the chronicity and after treatment. So that again is a challenge. Now, one of the biggest challenge is the data challenge. And that is because there are about 100 billion neurons which talk to each other using about 1,000 trillion synapses. Well, we have this inspiration of Human Genome Project, that we if we could sequence and map the whole human genome, we could map the human connectome too. But human genome only had 3 billion nucleotide pairs, and they were not as dynamic as the brain connections. So this data, even though there has been significant technological advancement, but the data of the brain it looks like this aspiration is ludicrous, that we cannot have this connectome. And there are some critics too who believe that this looking at each cellular connections in the brain is just waste of resources and time, and it's not going to achieve much. But then Human Genome Project has the same criticism when it was first conceived in late 80s or 90s. But what we have achieved there is remarkable. Now, with the advancement, you see these commercials all the time, that you can get your own whole genome sequenced, by ordering this from home. You can just send your blood sample and get the results in the mail in a few days with just minimal cost. And that's possible because of the success of Human Genome Project, which is like one of the scientific milestones in our history. And we aspire to have similar mapping or sequencing of our brain tissue too. And that's the eventual goal of neuroscience. And hopefully we will have it soon. And if you look at the innovations, the internet and all this information technology has developed in front of us, like within past few years, and the way it's making progress, it's not unacceptable to think about neuroscience making similar progress and giving us the whole each cellular connection and this whole map of brain in an individual. So there are enough reasons to be hopeful. Now, in the end, I think we will still have some time for questions and all. So in the end, I'm going to show you this slide, and this is just only 3% of human neurons and 0.0001%, like that's one of the 10,000 percentage of synapses simulation, how they talk to each other. And you will see how incredible the brain is. And what we are trying to achieve with this connectome project is looking at the whole brain this way and simulating it. So I will end my presentation here. This is just a brief idea of neuroscience, and in the next, I'm going to pick up a specific psychiatric disorder like schizophrenia and go through the neuroscience at different scale, from at the genetic level to cellular level to the circuitry level, and we'll see how those different scales are interrelated or interconnected. So yeah, that's it for me, and we can start discussions.
Yeah. Thanks, Dr. Ram, for the insightful presentation. I'm sure it was quite immersive visually. Um, Maybe over to Dr. Bhaskar. How do you want to take the questions?
Thanks, Ram, for the good,
Yeah.
wonderful lecture.
Thank you.
I would say we are just touching the outer surface of actually the multiplex and various whole brain organization. So for the introduction of the lecture, it is a very good one. Thank you for the wonderful deliberation. Now we are trying to form the questions, and let's see. Someone please help me with the question list. Here. How to go back? Yes. The first question that I would like to put here is by Dr. Paresa. He asked that as per new concept, mind doesn't exist. So what is your view on it?
Well, like you said in the beginning, I mean, if we can trace back all the functions of the mind to the brain, then there is no reasons to think about this concept of mind.
Yes. And the second one is actually a very interesting question. Dr. Dipankar Bagh has asked, can transsexuality be explained only by brain function? I would add homosexuality too, because what would be the same answer?
Well, that's hard to answer based on the empirical data. I'm not sure if there has been enough research done specifically on this, on these sexual preferences or no. But if I have to guess, I'm sure their brains are wired differently than the heterosexual individuals. But that's only my assumption, and I'm not speaking based on any empirical data or empirical results.
If I may add, actually, this is, as I said already, I'm a student of molecular neuroscience. So I have interest in various molecular neuroscience areas. So
Mhm.
what happened is in 2021, UK Biobank actually published one excellent neuroimaging and genetic data on same-sex individuals, and it is quite a huge number of data set, and they have found a relationship. They have found various changes in brain. If someone wants to know that in detail, that person can WhatsApp me or email me. I would send the paper in detail because I have the paper. I am not going into detail, but it is a very interesting question.
It
And
is, and again, I mean, I'm sure there are people who are interested in this topic and they are doing or researching further. But you talked of UK Biobank, and let me tell you, like, if there is a researcher here or interested in doing research, UK Biobank is a great resource to
yes,
use, and I mean, you can use it for free. You don't have to spend time collecting data, but you can get a lot of questions answered using that data set. So that's one great resource. I would
yes,
recommend that every if there's a new researcher or young resident or who wants to do something with the research, that's good starting point.
absolutely.
Yeah,
UK Biobank is doing very great.
yeah.
So uh, anyone from MediSquare, I would like to access the previous questions. I am getting the last 10 questions, but how I get the previous ones? Yes, got it. Sorry, me and my clumsy fingers. Yeah, there is one question from Dr. Silveraj. He asked, where does PET study stands in diagnostic certainty? I would like both Ram as well as Dr. Naren Rao to answer this question jointly.
Yeah, maybe Ram first. I'll
What, what,
follow.
Yes.
what was the question? I didn't get it.
Where does PET study stands in diagnostic certainty?
Or the pet studies? Well, so
Where does the diagnostic certainty of PET study? That's the question.
the pet studies are currently used in clinical practice in some cases, like in Lewy body dementia and Alzheimer's. If you have to differentiate, you can go for that uh, uh, dopamine transporters studies, and they are based on the radio tracers. The PET studies have been in use in some cases. In psychiatry, I guess, like with the better understanding, if we can use um, radioactive dopamine and glutamate and other neurotransmitters for diagnostic classification, it has a potential for better use or more use in future. But like I said, and again, uh, it's also been used in Alzheimer's when you're using this amyloid PET scan. Yeah.
Yeah, I concur that at present, if we are asking for a diagnostic certainty, it is mainly in the dementia. So none of the other psychiatric disorders, because I include dementia as part of the psychiatric disorder. Okay. So in dementia, both as simple as FDG PET has a better prediction compared to MRI. So diagnostic certainty wise, FDG PET, amyloid PET, and now the tau PET is upcoming, but the certainty of tau PET is not as high as the amyloid PET.
Yes.
So both FDG PET in advanced stages and maybe amyloid PET in some selected early stages of dementia, they both have very good diagnostic certainty. In other disorders, it's mainly in the research domain yet. So it hasn't translated into the clinical practice. Yeah.
Absolutely. And again, I mean, the neurotransmitter studies, especially in psychiatric disorders, they still need more research and observation before we can actually use the neurotransmitters in the diagnostic classification.
Yeah. So fair enough. Now there is another question that is also very interesting one. It is asked by Dr. N. He asked that direct animal models do not exist for any of the psychiatric symptoms. So how to understand the consistent neuropathologies from the animal trial?
That has been the challenge, that the anatomy of the brain in animals and humans are not as similar as, say, animal heart and human heart. And brain is the one organ which makes us different from all other animals. So that's a challenge, and that has been the reason, one of the reasons that the neuroscience research is lagging behind the other research areas. So and that's a very good question and very important challenge, that we cannot use animal models. We cannot make animals think and remember and feel and behave the same way as humans. So yeah, I don't know what the solution for that is. The solution probably maybe that we stop using animal models eventually and look for human brains using the best ways we can, like fMRI and PET and SPECT and other scans.
Okay, I would like something to add here because it falls in my area.
Mm-hmm.
Actually, there are three ways we can build different models of psychiatric symptoms. One is via animals. We are actually getting close to it. For example, CLANC3 gene deficiency animals, they actually show something close to the hyperactivity behavior of manic state. There is also another way we can see various brain models, that is via cerebral organoid or cerebroid. But again, cerebral organoid study has opened up a huge change in disease formulation. Let's say there is a professor of genetics as well as molecular neuroscience in California, Dr. Mootri. I am following his research for a long time. He started with forming cerebral organoids from various ancient human beings. Let's say Homo hominid, Neanderthal. Then he built Donovanis. Then he has built the early Cro-Magnons, and he has compared those brain organoids with brain organoids from current human being. And his team has recently published their research, I would say first paper of their research in Science, just few days ago. They have found there is a change of 60 key genes between the early human beings, let's say Neanderthals, and the current human beings, mean us. One of them is Nova1. And when Nova1 is mutated in normal human being, the brain structure changes. What happened in growing up from the early human beings, that is Neanderthal and to current human being, is we got a mutated Nova1 gene, and that changed our brain. It made our brain more rough, more convoluted. It increased our brain's shape, and ultimately it made our brain connect much more. Our brain's individual connections are much more, and it is actually a multi-dimensional view, which is not possible in early human being. So we have seen that by comparing cerebral organoid of different various human species, we are getting a huge difference. That is what making modeling of psychiatric disease so tough. And third one, what we are now attempting in all over the world is we are creating organoids of cerebral organoids from our patient's own brain. And we are trying to see individual organoid problems in a specific brain, which is derived from a patient. It is very early stage. It is very early stage. It is going to take at least, I would be very surprised if it goes by 15 to 20 years. It would take much more than that. But let's say by 30 years, we would see individualized disease modeling by forming cerebral organoid from pluripotent stem cell of a patient. This is how we are growing into various modeling for psychiatric diseases.
That's very exciting. Yeah, indeed.
Now, there is another question, and this is, I have seen so far, the best question is this. This is question by Dr. Ramesh Babu. Dr. Ramesh Babu has asked, what is the role of multi-scale neuroscience and concept of connectomy in understanding the neurodegeneration, number one, and understanding the traumatic brain injury and its evolution?
Absolutely. I mean, these are the few of the commonly studied areas in the multi-scale neuroscience, both at cellular and macroscopic or large-scale levels. And there have been all kind of, I mean, if you look at the neuroscience journals or high-impact neuroscience journals, they're full of different articles on both these subject areas. And like I said, overall, you can say that there is disruption in the connectivity at large scale, and there are cellular changes associated with the neurodegeneration and the TBI, which is seen in the postmortem brains. Again, I mean, this work is in progress, and eventually the goal is to develop or use these methods to identify or make a diagnosis or assess the treatment progress at the individual level, at the single subject level, so they can be used in clinical practice. We can say that we are not there yet, but what we are understanding about like the organizational changes with these disease conditions is improving with the time. So if we look at all the resting state networks and the way they coordinate with each other, that's disrupted in all disease states, including psychiatric and neurological diseases. The way they organized, which we learned by the graph theory methods, there are differences in the modular structure or the other quantifiable measures of those graphs, and so far most of these are at population level. The difference is when we combine a group of neurodegeneration individuals with the group of healthy individuals, we see significant differences. Now, to make it more clinically translatable is a work in progress for now.
Okay, I think, oh yes, there is another question by Dr. Pfizer. How does neuroscience helps in differentiating and understanding the concept of coexistence of seizure and pseudoseizure in a single patient?
Coexistence
of seizure and pseudoseizure, the network neuroscience view of coexistence of seizure and pseudoseizure in a single patient.
Well, that's very interesting and very difficult question to answer. Huh. Dr. Rao, you want to go?
Dr. Rao? Dr. Rao?
Yeah.
You are invited.
No, that's a very interesting question. So uh, what we need to understand is the brain doesn't work in isolation. So it constantly interacts with the environment, and then the existing pathology in the brain interacts with if there is any pathology outside, and then the interaction between the internal atmosphere with the external atmosphere brings about the abnormality. So there is no doubt that if somebody is having dissociative attack plus seizure, you will be able to see both of them if you actually do. There was a very interesting article a few years back which showed that the right parietal cortex abnormality in dissociative disorder individuals when compared to those who are normal. So we will be able to pick up, and especially if you club multimodality like video EEG and the MEG along with the functional MR studies, we will be able to differentiate both, and you will be able to get a rational explanation for that. So existence of one pathology doesn't prevent from another pathology being detected, if I have to put it in a nutshell.
Absolutely, yeah.
Absolutely.
Yeah.
So this ends the question answer sessions for us. We have to commit a few questions because we are running out of time. Thanks, Dr. Ram, for giving your valuable time, and
Thank
Dr.
you.
Narendra for coming here.
Dr. Bhaskar, you want to decide the best question? I think they have a cash award.
Yes, the Ramesh Babu and Dr. Faizar, these two questions are the best. Now, whom to choose? That's the duty I am giving to you because you are my senior. You take the blame for it.
I think both of them were very good question. But I think considering the topic of multiscale imaging from microscopy to the neuroimaging, I think Dr. Ramesh Babu's question aligns more with the multiscale theme of the topic. So I think it's a hard choice between the two, but I think we will give it to Dr. Ramesh Babu.
Okay. Okay.
Sure. I think we had a wonderful session. Thanks, Dr. Bhaskar, for the moderation and also very insightful comments. Thanks, Dr. Ram, for an excellent talk. And I'm sure everybody is looking forward for the next talk, which will be more related to the individual psychiatric disorder. And hopefully we will have a good audience the next week and an interactive session. Over to MediSquare for the further proceeding.
Thank you all for joining us today. We hope to see you again on coming Tuesday. Please block your date for 9th March 2021, same time from 8 p.m. to 9:30 p.m. Topic for our next webinar is Microscopy to Neuroimaging, Multiscale Neuroscience of Psychiatric Disorders like Schizophrenia, Part 2 by Dr. Ram J. Bishnoi. Also, based on many requests that we have received, we would like to inform you all that all our previous webinar archives are now available on our YouTube channel, Banai India. Do send us your feedback and comments, if any, on info@banai.org.in. Till then, stay safe. Good night.